Some look through this prism trying to hear the solo in a trio. With some genetic diseases, the cause seems to be an extra chromosome that interferes with normal development. One of these situations is Down syndrome. We're about to once again tread into the land of science fiction, become reality. One of the great pleasures of doing the Prism is discovering that I'm wrong about certain suppositions. Last week, I was speaking to Roger Reeves out of Johns Hopkins about a method he's developed which seems to have reversed Down syndrome in mice. Well, in the discussion, I suggested that it might be very hard to just turn off the extra chromosome because you need the other two. And he said, well, up to 6 months ago, I would have agreed with you. But now you should talk to Dr. Jeanne Lawrence. So, joining us now is Dr. Jeanne Lawrence. She's the interim chair and professor of cell and developmental biology and pediatrics at the University of Massachusetts. She's here to talk about, well, how I was wrong. Welcome, Dr. Lawrence.
Well, thank you for having me.
So tell me what you've discovered.
Well, what we have demonstrated recently is something we've been working on for 5 or 6 years, which is the idea that you can essentially silence one of the 3 chromosome 21s in Down syndrome patient cells. This is in a culture dish. But we found a way to sort of turn off the genes on that chromosome, basically correcting the genetic problem that leads to Down syndrome.
Now, we've already hinted about that in my introduction, but what is so important about chromosome 21?
Well, chromosome 21 itself isn't so particularly important, except that it causes this extremely common condition of Down syndrome, which affects about 1 in 700. So really, if you took sort of the top dozen common single gene defect diseases that we hear about, like cystic fibrosis or sickle cell anemia, and you added all those different diseases together, they'd only be about half as frequent as Down syndrome. So the 21st chromosome is important because it causes that. And it would be true of any chromosome that an imbalanced number of copies of the chromosome would cause problems. So for each chromosome, you get one from your mother and one from your father. So the normal state is to have 2 copies of that chromosome. But in Down syndrome, the individual has 3. And in fact, this arises commonly that there are these chromosomal number mistakes, but most of those fetuses are lost, they're miscarried spontaneously. So for example, if you had trisomy for chromosome 3, you might never even know you were pregnant because the fetus would be lost. In the case of trisomy 21, the chromosome is small, and that might be why it's compatible with life, because there's 300 or so genes on the chromosome, but it isn't like 1,000 genes on the chromosome. So it causes this very common condition, and because it affects so many genes, hundreds of genes, we really haven't had a lot of drive to try and figure out a way to correct the genetic defect, because it was like, how can you correct hundreds of genes? So if you want, I can just tell you very simply how we did that. I don't know if you want me to go into that detail or not.
Well, before we do, living in Washington, D.C., I've always been taught the triplicate is good. Why are having 3 genes so dangerous? Is it a dominance confusion?
Well, what it is that it's just a basic principle in biology that it's not just like whether you have the right sequence of a gene, like whether it's mutated or not. It also matters how much of the gene you have or how much you express. For example, if you have 3 copies of an enzyme that you're supposed to only have 2 of, then you could maybe have too much of a particular process in your cells that would make the cells unhealthy. So it's that the balance of your gene products is as important as whether the gene is exactly right or not.
So now let's move into your experiment. How were you able to suppress one without interfering with the other 2 that we need?
Well, the way we did this was to use a gene that I've been studying for a long time called XIST, X-I-S-T, that's on the X chromosome that is known to silence one X chromosome in females. And I can explain why that happens in a minute if you want. But So what we did was use a new genetic engineering technique. It's called zinc finger nucleases, but it's a way to sort of cut DNA and cut and paste DNA at the exact site you want. So we took this exist gene and we stuck it in a particular place on chromosome 21. And in this experiment, we only hit one of the 321s. We kind of designed our technique such that the probability of hitting all 3 or 2 was very low. We did have a few cells that hit 2 or 3 chromosomes, but very low percentage. And in the future, there are ways where that can be eliminated entirely. But anyway, so we stuck this one exist gene onto one of the 3 chromosome 21s and then turned this gene on, and it causes the silencing of the whole chromosome. In other words, the gene makes a product called an RNA that coats the chromosome on which the gene resides. So we only put the exist gene on one of the 3 chromosome 21s, so only one of them was silenced.
And when you did this and you cut the one chromosome that you wanted to silence,
Yep.
Did it then communicate to all the other replications that this is the proper order, copy me instead?
We did this in cells in culture. They were stem cells that were derived from a patient with Down syndrome. And so when we put the gene into one of the 3 chromosome 21s in a particular cell, then whenever that cell divides and replicates itself, that gene is still just in one of the chromosome 21s. So it's propagated throughout any cells from that point on.
Right now, is this experiment going to be more beneficial in terms of expanding our knowledge of Down syndrome, or is it really leading us towards the path of treatment?
So I think, the fact that you can take one gene and correct the overexpression of hundreds of genes now makes it possible to do things with both Down syndrome research and in the longer-term therapy that wasn't possible before. So for Down syndrome research now, we can use this to sort of understand, to figure out what is different about Down syndrome cells. So we can have the exact same cells in one culture dish, in 2 culture dishes, but in one of them we can correct we can silence this extra chromosome, and the other one, they're still trisomic, and we can ask what's different about them, what's kind of the pathology of those. And that will help right away to look for drugs that correct those pathologies. So that's the first sort of shorter-term impact. When you spoke to Roger Reeves, he was testing a drug in mice and trying to see if it mitigates some of the effects of how mice perform on certain tests and things. And so the mouse models of Down syndrome are very important, but they're not perfect replicas at all of human Down syndrome, right? So now we have human Down syndrome kind of in a culture dish, and the importance of our doing it in stem cells was because these stem cells can form different cells of the body. So we can make the stem cells form neurons or heart muscle or blood cells. And now we can ask for each of those systems what's different or wrong with the cells when they have the 3 chromosomes expressed. And then we can ask, does this drug reverse that pathology? So we can test it in a human system, in a culture dish. So it adds a whole other way to test whether you have a therapy that appears to be working. So anyway, that might be too technical, but just to explain.
No, I think it's very interesting, and it's obviously important because including a one-to-one model obviously is very helpful, because comparing to a mouse and comparing to a human, while it's not apples and oranges, is still different variations of fruit.
Yeah, it's different, and if you are going to try and treat people, you kind of want to be able to test what you're going to do as much as possible, and before you actually put it, expose people to it. So having a human system in culture is very valuable, and human stem cell biology is a very big growing field in science.
I think one of the things which excite me about this research, especially down the road, is not just about what we're doing here with Down syndrome, but about the potential generalizability of this kind of effort. Because I'm assuming there are multiple conditions, and you've intimated that there are, in which having that third chromosome or a fourth chromosome possibly even is particularly damaging. And if we figure out how to do it here, we probably can figure out to do it there as well, right?
Yes, in several instances. So the longer-term potential of this approach is that it's like a new type of genetic therapy. You could call it chromosome therapy. Okay, so we all know that genetic diseases are common and they're being heavily studied, but chromosomal diseases, because they involve many genes on the chromosome, we haven't really tried to apply genetic approaches to those before. We couldn't correct that genetic defect. So now we've shown that you can do that for Down syndrome. So there are some other disorders that are seen in live births. You're correct that this approach could be applicable to some other disorders like trisomy 13 and trisomy 18, also called Edwards or Pateau syndrome. Those occur in live births. They're not as common as Down syndrome, but they're devastating. The infants usually die within the first 1 to 2 years of life. So this same approach should be applicable to those conditions. In pregnancies, chromosomal abnormalities are really, really common because about 15% of known pregnancies spontaneously miscarry, and half of those are due to chromosomal abnormalities, half of them. But I wouldn't want to say that this technique is going to change that anytime soon because often those pregnancies are lost before you even know that the person's pregnant, okay? So I think that probably isn't going to affect this huge, massive miscarriages that occur right now. But there are other chromosomal abnormalities that this is a first step, okay? So it's demonstrating it for Down syndrome. It could work with trisomy 13 or 18. But going forward, I suspect my fellow scientists will start to apply different versions of what we've done to other chromosomal abnormalities. But I want to say it'll be a long time in the future before that could actually benefit patients living right now.
And this question probably also deals with looking into the future. But would this kind of research, would this kind of treatment have limitations in terms of windows of when it could be used?
Yes, I think, one of the things we're going to do now is work to take the Down syndrome mouse model. When you spoke to Roger Reeves last week, he may have spoken about the mouse models that he and others have been working on for a long time. And we're going to see if we can correct the syndrome of the mouse, the pathology of the whole mouse, but we're going to do that in a way that we can turn on this silencing of the chromosome. We can silence the chromosome at different times in development and see how much rescue of the pathology we get at different times. But I would expect that there will be certain aspects of Down syndrome that you have to sort of rescue earlier, and others could be later in development. For example, you may know that people with Down syndrome, now that they're living longer, we realize they with a very high frequency, get early onset Alzheimer's disease. And that's something that comes later in life. But also leukemia, there's a high rate of leukemia that is, in the young children. And the secondary effects of congenital heart disease might be approachable by a stem cell therapy. So we're at an early stage, but now we have these new possibilities in multiple directions to think, okay, how can we use this sort of whole new approach to mitigate different aspects of Down syndrome that affect different parts of the body.
Learning how silence in chromosomes may be golden. I'm Andrew Hiller for the Prism on the Voice of Russia, and I've been joined by Dr. Jeanne Lawrence. She's the interim chair and professor of cell and developmental biology and pediatrics at the University of Massachusetts. Thank you so much for your time and your work.
Well, thank you. I appreciate your interest. And, if you want to look at science fiction, I think there was recently a paper describing, building a heart out of like a scaffold and stem cells in a dish, So this is the whole idea of regenerative medicine, where you can use cells to help treat the body.
Yeah, I actually had an interview not too long ago. I think it was from someone from New York University who had developed a laser 3D printer which was using embryonic stem cells and he's gotten it to the point where it can build structures, and he thinks within a year or 3 years he'll be able to have it develop organs entirely. It's mind-blowing to me.
Oh, so that's cool. So I need to find out the name of that person. I'm reading this literature and trying to find out. I would like to collaborate with someone like that because, the importance of what we did was also that we did it in stem cells.
Oh, shoot me an email. I'll try to dig him up.
Okay.
And I'll at least get you his name and basics.
Oh, yeah, and then actually just somebody, I said to my colleague this morning, we need to get the name of the best person to collaborate with, and she said, I saw someone who's actually doing a 3D printing of tissues or something. Sounds like, so she was going to look for their name.
Okay.
So anyway, I appreciate your interest